Our next speaker is Dr. Kinga Hosszu, who is the co-lab director of Memorial Sloan Kettering's Cancer Center and the Immune Discovery and Modeling Service in the Department of Pediatrics. Dr. Hosszu received her PhD in genetics at Stony Brook University, after which she completed a postdoctoral research focused in the role of STS protein in the regulatory T-cell development and function and receptor complex formation during C1q-mediated monocyte to dendritic cell transition. Dr. Hosszu has been the lab co-director at the Immune Discoverer and Modeling Service since 2020, where she utilizes state-of-the-art technologies to help improve HSC transplantation by exploring how individual differences between patients impact the efficacy of the therapies. Today, Dr. Hosszu is going to tell us about her work establishing and optimizing a translational program of immune monitoring for biomarker discovery. And I'm learning that she is a longtime supporter of MetroFlow, attendee. Maybe you could tell more about that. Thank you.
Thank you so much. Thank you for that introduction. Sorry, I have a water. I just heard no drinks, but it's allergy season for me, so if I don't have a water, for sure, I will break out in a cough. But thank you so much for coming to my talk, and thank you for inviting me to this talk. It is special because as a graduate student, MetroFlow was the first meeting that I ever attended, and now I'm here sort of presenting my work to you, so it's a full circle moment here for me. But I'm really excited to talk about the work that we do at Sloan Kettering in the lab, where we're really trying to answer one fundamental question. How does the immune system reconstitute after transplantation, cell therapy, CAR T-cell therapy, and other immune treatments in cancer patients? And today, I will talk to you about how we established an immune monitoring program specifically for allogeneic transplantation patients, and how we optimize this translational program for biomarker discovery. Oh, nope, it's not going. I'm pressing the button and it's not going forward. No. I pressed it many times, so if it starts to go, it might go real quick. It's gonna be, thank you so much, it was great talking. Now it's dark.
I think you might be reset.
Okay, thank you. But maybe while it resets, I can just sort of foreshadow. I will sort of divide this presentation into three. And first, I will introduce to you why we immune monitor and what is immune monitoring. And then I will introduce how we built an immune monitoring program. And then I will give you some example of how we turn color, spectral flow cytometry data, into comprehension, into interpretation of results. So first, before we go into establishing the immune monitoring program, the question is, why do we immune monitor? Why follow the immune system after transplantation, and what kind of insight can it give us? The short answer is that immune reconstitution for transplant patients is everything. For these patients, the immune reconstitution will give us an idea of response to infection. It will give us an idea for risk for graft versus host disease, risk for relapse. All of these are complications, negative outcomes for these patients. So monitoring the immune system and establishing the patterns of reconstitution, how dysfunctional it might be, is super essential for these patients, for the clinicians, to be able to identify the risks and intervene early. And specifically today, I'm talking about our lab monitoring allogeneic hematopoietic stem cell transplantation, or from here on, I'm just going to call it transplant, and you and I know what I'm talking about because it's too long. But our lab monitors immune reconstitution in multiple treatment modalities. So today, I'm just talking to you about transplant because when we established the lab, in 2020. That was the purpose of the lab, to work with the bone marrow transplantation program, specifically in pediatric patients, and monitor every single patient that comes through Memorial Sloan Kettering through that program. So these patients who get allogeneic transplant basically receive donor stem cells, so from another person, to restore their immune responses. And it's a potentially curative modality for malignant and non-malignant diseases, but it does come with its own immune challenges, because before the treatment is given, the patients have to go through lymphodepletion to deplete their immune system. So it makes space for the new immune system to recover. And this lymphodepletion can cause some damage in the host tissues and will result potentially in delayed immune reconstitution. Once the patient gets the cells, then there is a whole other set of complications or negative outcome that can develop. Some of these are graft versus host disease, infections, relapse, graft failure, and basically the whole point of looking at the immune system in detail after transplant is to figure out if we can predict complications early and hopefully prevent them. So after these patients get the cells, there is a very predictable pattern of immune recovery that you can follow. And first, it's always the innate immune system that recovers. Right? Maybe? Yes. The innate immune system that recovers, and you see monocytes, neutrophils, and NK cells recover within 1 to 2 months of transplant, followed by CD8 positive T cells and B cells, and then last to recover are CD4 positive T cells. And ironically, CD4 positive T cells are the most important readout for a transplant patient because it's truly a clinical readout. There is a lot of data out there showing that a faster and more robust CD4 T cell recovery results in the patients getting better outcomes, so not getting complications of graft versus host disease or infections or relapse. Conversely, if there is poor or delayed immune recovery, it will increase the risk of complications, relapse, and infection. So how do we monitor these patients, and what is immune monitoring? It sounds fancy, but at the core of it, it really is a very simple idea, and it's kind of self-explanatory from the expression. So we are systematically measuring the immune cells and also secreted protein dynamics over time. The purpose of it is really to understand how the immune system recovers, so immune reconstitution, how the cells might show dysfunction, and how the patient will respond to therapy based on these immune patterns that we observe. And here on this figure, you can kind of see the major steps after transplantation, but also under this CAR T-cell therapy as well, because I mentioned that our lab does monitor both types of treatments, although today we're focusing on transplant. And here, you can see in the red arrows the time points that we have selected to collect samples. So there are a lot of them. Already before the therapy, we collect some samples, and then once the patient gets the infusion of the cells, we collect at very specific time points for 2 years after the treatment. And if you just take a bird's eye view to figure out how does immune monitoring look like, it's a very basic idea. You collect these samples, and you can do 2 things. You can do an immediate assay, or you can isolate the plasma and the PBMCs, freeze them, and then later do some kind of more discovery type assays. Sounds simple, right? So not so simple. It's a little bit complex when you actually start looking at the details. So let's tease it apart a little bit. When you perform immediate assays, usually your trying to look for an answer that can guide clinical interventions and improve outcomes in real time. So immediate assays are usually done for clinical purposes, and there are multiple types of assays that can be performed. A major part of it is flow cytometry, when they're looking at cell subsets that develop. But also there are functional assays looking at cell activation and cytotoxicity. They're measuring drugs, especially the lymphodepletion drugs that were given before transplant, to identify their PK/PD patterns. They can look for antigen expression or TCR sequencing and HLA typing to figure out if the cell is a host or a donor origin. And there are several protein assays, including IgG testing, to figure out antibody responses. So all of these are pieces of a puzzle that the clinicians can look at to identify if the immune system is recovering as expected or if they would have to intervene early. On the other side, there are the frozen cells, and usually this is for discovery analysis, so for research. When you can basically take a bigger batch of the samples and throw bigger assays at it and try to find new biomarkers or gain novel insights into the mechanism. Again, there are many assays that you can technically perform, but the big part of it is multi-parameter flow cytometry, when you're trying to do really in-depth immunophenotyping and find new types of cells or cells that express very specific cell surface markers. But other than that, you can perform functional assays and look for cytokine secretion. You can still perform PK/PD studies. There are many drugs that are novel and don't have a clinical test, so we do a lot of those in my lab. You can do imaging for cancer topology, transcriptomics for establishing transcriptional profiles, and one of the really hot interests for transplant patients is looking at the microbiome, because there's a lot of data associating specific microbiome patterns with specific complications or lack thereof in these transplant patients. So that's a hot topic that I will not talk about today, but I'm very interested in that area as well. But whether you're monitoring for clinical purposes in an immediate assay, or you're looking for research analysis in a discovery assay, at the end there is data analysis and data interpretation. So I did mention that flow cytometry is a big part of both of these steps because it serves as an easy and predictive tool of recovery and risk in these patients. It can identify the pace and balance of immune reconstitution. It can show us how the thymus recovers after transplant by monitoring the newly created T cells called recent thymic emigrants, of which I will talk a little bit about at the end of this presentation. You can look at the patterns of T cell maturation by naive, memory, and effector dynamics. You can also look at homing markers and identify some kind of trafficking patterns, or look for a wider array of predictive markers for immune signatures. So flow cytometry is an awesome tool. I'm preaching to the choir here. I know you guys are all on board with this. So now that we established why immune monitoring happens and what immune monitoring really is, let's dive into building an immune monitoring program. And I will heavily rely on what we established at Memorial Sloan Kettering. Of course, that's my experience, where we really try to establish a program that is robust, reliable, and dare I say, collaborative. So what is the mission of any immune monitoring program? It is to build a robust and standardized workflow that can support both a clinical insight and also new biomarker discovery. So depending on how big your immune monitoring program is, how big your infrastructure is, how many people you have, how much money you have for this, you can focus more on clinical or more on research. At MSK, we are very heavily research focused. Of course, our immune monitoring does have a clinical component, but we do have a huge research arm of our immune monitoring, and that's what I'm going to focus on today in more detail for you guys. We focus on research not because we have a lot of money, but, but because we're surrounded by a lot of very smart, curious people who want to know, and somehow we always find the money by miracle. But what are the essential components? So before we even get into flow cytometry or whatnot, how do you even establish one of these things? So at the heart of it all sits an MU monitoring hub. And for this program, it's our lab at Sloan Kettering. But this MU monitoring hub basically directs the entire workflow, networks with everyone, and keeps track of the movement of the samples, the data, analyzes the data. So what are the major components of this? Basically, there is a standardized sample pipeline, starting with establishing an MU monitoring schedule. So when we started the lab in 2020, that was my first job, established the MU monitoring schedule, and I was an immunologist, new to the cancer and transplantation world. So I had my work cut out for me to do my homework real quick and figure out what would be the interesting time points, the really essential time points to collect samples. But once we establish that, then we have to liaise with the clinical team because they have to identify patients, they have to collect the samples, they have to make sure that they hit every time point for each patient. And as you saw in one of the previous slides, there's a lot of those time points where we collect samples. So after the sample is collected, it has to be transported, and it sounds trivial, but trust me, it's not trivial to get a sample from one place to another in a hospital. Once it gets to the place where the samples are going to be processed, we isolate plasma and PBMCs there, and it has to be stored. So you need kind of a big enough infrastructure to look ahead and be able to store thousands of samples over the course of years from hundreds of patients. But once you've kind of gotten to that point, there's the next step of assay development and validation. And even though what I'm talking about to you today is a research workflow, so we're not really bound by clinical constraints, at the end of the day, we need to create reproducible results across patients, across time points, across cohorts. So our validation and our criteria ended up being almost clinical-like. Just establishing all the SOPs, all the workflows, training our technicians, calibrating the instruments, which has its own point, but it kind of goes into this step. All are very rigorously done in our lab, rigorously documented, so that we can trust the data at the end of the tunnel. And then we enter the data pipeline. So once you've done the assays, you generate a lot of data. And so now what happens? The data actually goes on a very centralized trajectory after that point. And of course, we have to analyze the raw data. So you get raw flow cytometry file. We have to get some frequencies and intensities. And we also do some proteomics assays as well. So we have to collect that research data too. And then you have to put it together with the clinical data. For all the patients who we have samples for, we need the clinical parameters, we need demographics, and let me tell you, it was a surprise for me in a hospital, but that's the harder part to get. The data that's already there, it's much harder to collect, and it has to be kept up to date periodically, because obviously the patients get new and new complications as time goes on, if they do, so it has to be constantly updated, and that's very hard. But once you kind of figure out how to do that and you put all of this together, you kind of go into statistical analysis where you're trying to figure out correlations between your clinical parameters, between the patient outcome, whether they got GVHD or not, whether they relapsed or not, and your research readouts. So all these flow cytometry analyzed cell subsets or the proteomics data, or a combination of them to maybe establish a biomarker signature. Once we kind of get there and throughout all of that, the data is housed in a curated database where people, dedicated people, have to upkeep the data, collate the data together, and update the data in a very periodic manner. And our lab kind of sits in the middle, like I mentioned, to sort of perform also the governance and collaboration steps. So not only do we keep the SOPs in our lab officially, but we perform a lot of the training for this entire hub network. We take care of data sharing agreements, and we are also the data analysis and publication center for our data sets. So it's not a trivial undertaking. We, in essence, do two types of main assays. I did mention that we do some proteomics, so I threw it up here even though you're not interested, you're interested in the flow cytometry more, but I just want to illustrate that it's really two arms of the immune system that we look at. We use Olink proteomics to analyze secreted proteins. It is a great discovery tool that I didn't know about before 2020, and now I love it. So if someone doesn't know what kind of immune changes are happening in the secreted world of cytokines and other immune proteins, Olink is a really good tool because it measures a large number of proteins in a small amount of sample all at the same time. So that aside, we also do a lot of flow cytometry. And because we're working with pediatric patients after transplant, so as I mentioned, they get lymphodepletion. It means that early on, after transplant, they basically have no leukocytes. So it's very hard to split up the sample into multiple parts. So we had to design these really big flow cytometry panels, and we use spectral flow cytometry for that reason. I also didn't use spectral flow cytometry before 2020, so this is like all my moments of confession to you guys. But I have to say that once I started using, I wouldn't go back. So if you haven't made the switch, I highly recommend checking out spectral flow cytometry. But we use a Cytek Aurora for all of our flow cytometry needs. Okay, we got stuck almost. And for immune monitoring in the transplant patients, we use 3 pre-developed panels. We developed these in my lab, and I will talk a little bit about how we developed them. So in case you're yourself developing a panel and you're curious, I will tell you some tips or tricks. But essentially, we're looking at T cells, dendritic cells, and B cells at all these time points in all the patients. The reason being is when I started at MSK, most people were interested in T cells. They are the effector cells in cancer. So that's the most immediate cell that everybody wants to learn how they function. I'm interested in dendritic cells, as Thomas introduced me in the beginning. My graduate work was in dendritic cells. I think they're awesome. So that was my initiative with a few other investigators at MSK that I would like to look at the dendritic cells also. And then we also look at B cells because at the end of the day, they're going to be the ones who mount the antibody responses. And so we wanted to see what are the patterns of B cell recovery as well to get a full picture of the immune reconstitution. But I will focus a little bit on this T cell panel, and I will tell you the 4 commandments of flow panel design. You probably already designed flow panels, hence you're here at MetroFlow. So I'm not going to go into the basics of how to design it. There are a lot of good tools out there, a lot of good educational tools, even just on YouTube. Even the vendors of antibodies have a lot of good tools to build these big panels. So I'm not going to go into the little basic nitty-gritty details, but I will tell you the 4 things that I would have liked to hear from someone when I started building these big panels. And the first being, start smart. So if you can find a pre-developed panel that will suit your needs, use it. There are no prizes for developing a flow cytometry panel from scratch. And you can save a lot of time for yourself. Nowadays, there are actually a lot of vendor-developed panels that are validated, and I think it's awesome. When I started in 2020, not so many. Most of these pre-developed combinations of antibodies were for clinical purposes. So we had to embark on developing our own combination, but we used an OMIP. And so OMIPs, as many of you know, are optimized multicolor flow cytometry immunofluorescence panels that are published in cytometry. And they have all the information that you would ever need about a panel, you know, which antibodies to buy, how to analyze it, how much antibody to use per cell number. So I think it's a great starting point. Or if it suits your complete needs, then don't even, you know, tweak it. But basically, when we started out, we already knew that we have to measure T cells. We're interested in exhaustion, inhibition markers, activation markers, and cell maturation from naive to memory to effector type cells. And this panel already had all of them in 28 different colors. So we started out with this panel. And before I kind of showed you a schematic, so now I took that schematic, that's our final panel. And this, I covered up the things on the panel that we have now that were not on this OMIP. As you see, most of the things were already on the OMIP. And basically, we were able to identify CD4, CD8s, and T regulatory cells and look at the exhaustion markers on them, a lot of activation markers, and cell differentiation as well. So this was already a really good start. But then my next recommendation is collaborate early. Especially if you're in an environment like I was, where you're not only going to be doing your own research, but because you develop this platform, a lot of people will want to work with you and use your lab as a platform for their research. So I already knew from the beginning that there's going to be a lot of collaborators who are interested in working with us, and we talked to all of these collaborators early on to kind of get feedback from them for what are the markers of interest for everyone. So we then proceeded to change this panel based on all this information. And I just put in here some publications. Some of it came out later based on work that we did with this panel. And we sort of systematically started putting things on that are indicated by the red arrows. So slowly the panel built itself, and we got to the next step. And so that recommendation is design your panel flexibly. If you've ever collaborated with someone, you might have had this experience where your collaborator comes and says, you have this beautiful tool, in my case, a big flow cytometry panel. I have this protein of interest that's not on your panel. If you could only put this one protein on one antibody on your panel, we would love to collaborate. And so over time, we realized that the best way to do this is to create space in this panel, some flexible markers that can be swapped out to custom antibodies of interest. It might come as like something to you that you're already doing. It came as a revelation for me, so I wanted to put it into the commandments. And so what we did, essentially, the last part of the panel that we added on is these new T cells, recent thymic emigrants. They are so important for transplant. They're not so important for some of the other cancer therapies that want to measure immune reconstitution. So we can swap these out, and we made those two markers PE and Alexa 647, so that you know that pretty much any antibody in the world is going to be available in those colors, and you can just swap them in, and you don't need to change your entire panel just to accommodate one new protein of interest. We also did this little dump channel for CD14 and CD15 for the neutrophils and the monocytes. The purpose is we're already working with cancer patients, so everything is already a little bit messy. There's a lot of background binding, and so any background that we can eliminate with confidence is a good win, a good step. So after this, we get to the last step, hopefully, and that's to evolve strategically. So once you have a flow cytometry panel, especially now it's a big flow cytometry panel, right? And it's informative, you like it, it works. It's very tempting to say, I'm gonna lock it in and I'm never gonna change it. But data is constantly evolving. People are publishing new results every day. And you're going to find new proteins that are probably interesting in your field of interest. So how do you change the panel, right? Our approach was to not do it reactively. So every time someone comes to us and says, yeah, this is the most important protein that you've never heard of, usually I ask the question of why. Why is this important? Is this important for you, for your project? Is this a niche protein? Or is it something that may be of interest to everyone in the transplant community? If it's the first niche interest, then we go to step 3, the flexible design. We swap it in, we swap it out later. It's for that project, we are able to measure the protein. But if it's of interest to the larger community, then we want to evolve strategically, and we will add in these proteins. And so we recently did that for 2 things, where we wanted to identify T follicular helper cells and also a very specific regulatory T cell type. So we added on 2 more proteins very recently to arrive at our final panel of interest for T cells. We did the same thing for dendritic cells and B cells, but I wanted to focus on the T cell panel because that's everybody's favorite panel at MSK, because everybody is interested in T cells. So now that we've gone through the 4 pillars, I just wanted to quickly touch on 3 additional things that in the lab is very helpful for us. So obviously there are best practices for flow cytometry, but at the same time, I think it's very important to be practical and maybe not perfectionist, to arrive at a point that is good enough and not necessarily try to push perfection further. And so one of the things that we did this with is single stain controls. You need single stain controls to unmix your spectral flow cytometry data, and best practices recommend that you create single color controls for every single one of your experiments fresh, right? But we run 5, 6 different panels every day in the lab. Each of them have like 20 to 35 antibodies. So that would be pandemonium every day. So what we do instead is every single time we get a new lot of the same antibody, we create a new single color control. We will record it in the single color library with the lot number, and then we use the lot matched unmixing approach in my lab. This was very helpful. Over time, we realized that it does not create unmixing issues if you're on top of it. The other thing that we do is fluorescence -1 controls. That's the other best practices for each one of your antibodies on the panel. Create a fluorescence -1 control, FMO control, so that you can gate with confidence. But again, think 5, 6 panels, 20 to 35 antibodies a day. If we did an FMO every day for every single one of our panels, we'd have to open a side gig and flip burgers to just cover our FMO business there. So we decided that during panel development, we identify key antibodies where the pattern of positive and negative expression is a little bit wishy-washy. Your populations are not clearly negative and positive where you can comfortably gate. So for all of those antibodies, we create an FMO for every experiment. For everything else, we just figure out how to gate by field. And the last thing on this list is bridging controls. So instead of cutting down on work, we added work with this one, but it didn't add a lot of work in the end. So at some point we thought back and we said, we use the same panel for multiple projects, not just the transplant projects. We create a lot of data sets with this panel. So maybe in the future we would want to combine them. How can we combine them? So we created a bridging sample, and we run the same sample with every single batch of every experiment that we do with the same panel. So for the T cells, we have like a zillion aliquots of Jurkat cells frozen down, and we just thaw that one aliquot of the same passage number of the same lot of that Jurkat cell with every single one of our batches, and this allows us to bridge batches of samples together. So now we went through the building of an immune monitoring program, and let me touch a little bit about how we turn color into comprehension, how we look at the data and interpret it or analyze it. And just want to be conscious of time. It's perfect. So just to illustrate the magnitude of the data that we have to sort of face, or faces us, for each patient, I mentioned the 3 flow panels. That generates 250 parameters with manual gating. Now, some of you might be thinking, but Kinga, why are you doing manual gating when there's algorithm-assisted analysis? And just give me a minute, I will touch on it in the last slide, but just bear with me, we do manual gating for every single sample. 250 parameters at least, right? And remember that O-link data that I talked about? So we run 2 panels at every time point. That's 190 parameters. So add that together. That's 450 parameters per patient times 10 time points. And that creates 4,500 data points per patient. And that is what ChatGPT thought it looks like for one of our lab members. So that's a lot of data, but we didn't stop there. It's not just our data. We actually are part of a consortium of 6 transplant centers, and every single one of those transplant centers monitors immune reconstitution the same way as we do. We harmonized our workflow. We harmonized how we collect samples, what patients we collect samples from, what tubes we use for collecting those samples, how those samples are frozen down and thawed. We harmonized our antibody panels. Everybody bought, I don't know if Cytec is here. It's not an advertisement. I just love the Aurora, and everyone bought a Cytec Aurora at all of these 6 institutions for this purpose. And basically, that allowed us to harmonize the data creation. And we even have a data analysis cookbook that lets everybody be guided how to gate the samples, which I will introduce a little bit of it in a second. So all together, these 6 centers are sharing their data, and we're all analyzing a really large amount of data for a big number of patients. And there is a reason to do this. It sounds like already at MSK we have a lot of patients, but if you really think about it, these patients are such a heterogeneous group. It's not like cell culture and you generate n equals to 8 and you're already like really good shape. Unfortunately, each patient is a little cosmos in themselves. There are female and male patients, patients who have malignant and non-malignant diseases, patients who got they're different age. They go from age 0 to 26 in a pediatric young adult transplantation world. You also have at least 3 different transplant types included in this analysis. So there's cord blood, T-replete, and T-cell deplete transplants. There are different donor types. They can be a different relation to the patient. And then all these patients develop different complications, different outcomes. So think it's a lot of different versions of transplant patients that go into this analysis. And the only way to overcome that is by numbers. So combining multiple data sets, more patients, allows us to be more statistically sure of the results, allows us to do multivariate analysis with a number of these categories so that we can figure out what are these, which one of these demographic or clinical outputs is associated with one of the research outputs. So we do have our work cut out for us, but we do love it. And so let me tell you a little bit about one example. So recent thymic emigrants, they're these progenitor T cells that are just coming out of the thymus. They are not naive T cells yet. They take about 3 weeks to become naive T cells. And you can measure them after transplant by flow cytometry. And the reason why you want to measure it is because as you are giving a transplant stem cells to these patients, this new immune system has to develop, right? Recover. And in order for the T cells to recover, these stem cells have to go to the bone marrow, to the thymus, and the thymus has to create these cells. So by measuring the RTEs, the recent thymic emigrants, you're essentially measuring thymic function during transplant. And you expect these RTEs to come back, reconstitute after 3 months after transplant. And you can distinguish them by flow cytometry by looking at naive CD4 positive cells that express CD31 on their purpose. So those are recent thymic emigrants. And what we did between the consortium members is that everybody bought a commercial PBMC sample. As you see, we bought it from Stem Cell Tech. And everyone bought the same lot of this PBMC, and then we threw our flow cytometry panel at them, at each center, and then we followed this gating path. So to arrive at recent thymic emigrants, as you probably already guessed, we just go through regular, gate out debris, gate in just single cells, gate out the doublets, and then we want to gate out any dead cells and also anything CD45 negative. In this sample, you don't have a lot of CD45 negative cells, but in a cancer world, in the transplant world, a lot of the times, the more tumor burden the patients have, the more CD45 negative cells we might have. So it's very important to gate on leukocytes alone. And then we gate on the T cells by CD3. After that, we actually gate out the TCR gamma delta cells so that we arrive at the majority of the alpha beta T cell receptor T cells. And then CD4 positive cells. At the moment, we can't identify recent thymic emigrants by flow cytometry in CD8 positive cells because the same markers don't work the same way in CD8 cells. So we only assess CD4 positive cells, and then we look at the naive T cells by CD45 RA expression, and then anything that has CD31 is a recent thymic emigrant. And basically at every single one of these consortium member transplant center labs, they had to go through this gating on the same sample and show that each of these gates had very similar frequencies of cells. Once we did that, we combined all of our data. And here you can see days after transplant. This is our combined data from 4 centers, not 6, because 2 centers didn't have data ready to contribute to this analysis. But as you see, their trajectories of lowest curves really overlap, which made us confident that the harmonization of all of these parameters really worked. And we were confident in analyzing the data. Now, of course, we analyzed these data for a lot of things, but here I'm going to show you just one quick example of looking at graft versus host disease. And basically, graft versus host disease is when the donor's immune system, or immune cells, which is the graft that you give the patient, mount an immune attack against the recipient tissues or the host. Also generated by ChatGPT. And we analyzed, we split up the patients into 2 groups. So in the blue dots, you can see patients who either didn't have GVHD or had really low-grade GVHD. And in the red dots, you see patients who have high, severe grades of GVHD. The first 3 months after transplant, nothing happens. No RTEs are found. But once we look at 6 months, you can see the blue patients who don't have GVHD or really low-grade GVHD already have RTEs coming in, while the patients with the severe GVHD have no RTEs. And this actually lasts for up to 2 years, as we measure it all the way to 2 years. The patients with severe GVHD don't have RTEs, or they're slower to reconstitute. Why? Our hypothesis is that GVHD itself, which causes tissue damage in the patient, attacks the thymus, causes thymic damage, and therefore the thymic output, the thymic function is affected and reduced. Of course, we have to look into it more, and there's a lot more that we're analyzing, but this is very interesting data already. And last, I would like to touch on one last question of why, Kinga, why are you not doing algorithm-assisted analysis? We do. We actually do a lot of it, but it's not our standard workflow. Our standard workflow is manual gating, but on the side, we also do algorithm-assisted analysis as a discovery workflow to discover new cell types or co-expression of markers on the existing cell types that would be associated with outcome. We don't do it for a mainstream workflow because not only do we work in batches, but we work in several different institutions. And if you've done any algorithm-assisted analysis, you know it's so sensitive to batch effects. And it will tell you that you have a whole new cell type in this one group of patients just because of a batch effect. So we only use it for batches that really match each other, and we do this discovery analysis, and whatever result we might get out of the discovery algorithm-assisted analysis will guide our manual gating. So in this case, we were curious if before the onset of GVHD, before even clinically you can identify that the patient will have GVHD, are there any cell types that would be associated with the development of GVHD? Again, we split up the patients into 2. The green patients are the ones who will not get GVHD or very low grade, and the red patients will get really severe GVHD. And with the algorithm-assisted analysis, it identified 11 different populations, 11 different clusters in our sample set. And we wanted to look at cell types or clusters that are significantly different between the two groups of patients. We identified 4. One of those were recent thymic emigrants, so good job confirming our manual gating analysis. But we also identified 3 more, and these are effector and memory type cells that have very specific homing markers. So that's a next step in our analysis, to go and manually gate those cells in all the consortium samples and figure out if this algorithm-assisted analysis will stand in that analysis as well. So with that, I conclude how we turn color into comprehension, and I hope that I was able to give you a little bit of introduction of what MU monitoring is and how we establish an MU monitoring program. And if you are developing a flow cytometry panel, whether it's for MU monitoring or not, maybe you can use some of the commandments of panel development. And thank you.
Thank you so much. That was a great overview. I appreciate it. Maybe we could do a couple of questions. Awesome. Sorry, I'm going with closest first.
Morning, Joe.
You jump over there.
Thank you. Great presentation, very lucid, and kudos to you for undertaking the harmonization effort. And just something to pick from harmonization. If you are seeing the exact same pattern of gating after harmonization across all 6 centers, so why are the high-dimensional algorithms not able to pick and homogenize the data sets? Could you comment on that?
Can you, sorry, because it's hard to hear, can you just repeat the question?
Sure. So since you are undertaking the harmonization effort across all 6 centers,
Yes.
and then collating the data, so why are the high-dimensional clustering algorithms not able to collate the data and give you similar islands? And why do you think you are identifying different populations?
So that's a great question, but even if you're running samples in 2 batches in your own lab, you can get batch effects, right? Sometimes we know why the batch effect happened, and sometimes it's just the phase of the moon can create some batch effects. Now imagine this from like different centers, but the main reason is because our colleagues at the 6 centers, while we use the same base panel, they changed out some of the additional markers. So they're not able to combine it in algorithm-assisted analysis.
Okay, and can I ask another question?
Maybe we'll go to Zoom. Sorry, thank you.
Hi, we have a question from Zoom. Is there age difference in patients with graft versus host disease and non-graft versus host disease? Because the thymus shrinks with aging, which might impact the number of recent thymic emigrants.
Yes. So I didn't include a lot more of the RTE analysis, but there is a lot of information out there. The thymus basically decreases in size with age, and after the age of 10, you actually only have 80% of the thymic mass. So we split up our patients into before and after age of 10, and we were able to confirm that the patients who are older have less RTEs. They will not reconstitute to the level as the younger patients do. And for GVHD, there might be age-related differences, but the most important differences are across transplant types. So patients who get cord blood will get a lot more GVHD. So in this combined analysis, without teasing apart which transplant platform you're talking about, it might not be so obvious with the GVHD.
Very nice, thank you. So in the interest of time, we'll move on to our next talk, but I saw there were a lot more hands, so maybe...
Feel free to find me.
Awesome, thank you so much,
you.
that was wonderful.